Chalo ji, to start with, what is asexual reproduction? Anyone? One mark question. What is asexual reproduction?
Sir, a type of reproduction in which only one parent is involved for the formation of the new generation.
That's slightly wrong answer. Not fully correct, I'll give half out of 1. When we started reproduction, that time I'd given the right definition for asexual reproduction or sexual reproduction. See, there are few species are there. So in those species, let's say the female can change its gender, become a male, and produce both the offspring. Means it can produce sperm and egg, and it can produce a offspring. Will not fully call it as asexual reproduction. So in asexual reproduction, we say one parent is involved, but that doesn't make the definition of asexual reproduction. That makes a characteristics of asexual reproduction. Anyone?
So there is no exchange of gametes?
There is no exchange of gametes, or we'll say there is no fusion of gametes, or gametes are not involved, right? Reproduction in which there is no fusion, there is no fusion of gametes or zygote is not formed, is asexual reproduction. So you'll say reproduction in which gametes are not involved or zygote is not formed is called asexual reproduction. So what are characteristics? Only one parent is involved. What else?
Sir, not much variation in the progeny or the new generation.
Okay, we'll not say not much variation. Anyone, what is the right word for it?
Is identical to the parent.
Very good. Offspring are identical to the parents. And what's the scientific word for offspring being identical to the parents? Clones of parents. Offspring are clones of parents. So there is no mating or fertilization. No genetic evolution. Right? जब कोई genes involved ही नहीं है, genes are identical, so there is no genetic evolution is there. So first type of asexual reproduction. Anyone?
Binary fusion.
Binary fission. So what is binary fission?
So nucleus divides into two parts. One is so it divides into the two parts, producing offsprings.
When nucleus divides in two parts, producing offspring. Okay. Then what's the difference between binary fission and mitosis? Why we can't call binary fission as mitosis? So I think what you have answered is partially or mostly correct. Anyone else? What's the, let's take this one first. What's the difference between binary fission and mitosis?
So in binary fission, the parental body nucleus is divided into two, but in mitosis, any particular cell is divided.
So binary fission also takes place in unicellular only. And in unicellular, the parental cell divides into two. And same thing is happening in mitosis. The parental cell is divided into two. Anyone? I've changed the question now. So it's a 2 marks question. Write two differences between binary fission and mitosis.
So when in binary fission, the offsprings are produced, they eventually grow nucleus, but in mitosis, there is a nucleus present in them after division.
No, it's not like that. In binary fission also, the nucleus is dividing and then the full body is dividing.
So the offsprings have the nucleus at the time of division as well.
That's true. In binary fission, it's not like that the body is divided into two parts and then the second part is developing the nucleus. See, it is the clone. And when a cell is a clone, that means that they have got exact set of DNA and RNA. That's what clone means. Whether it's a small body or a big body, but exact sets of DNA is there. So in fission or in mitosis, both ways, first the DNA gets divided into two parts and then the division takes place. So both of them are more or less same to each other, but they have to be some difference, right? So 2 marks question, what's the difference between binary fission and mitosis? I'm waiting. Kirti, Aditi, Arohi, Abhiraj. Let's see who is answering first. Okay, can you give me an example where binary fission takes place?
Amoeba.
Or
Sir, leishmania.
and
Sir, binary fission takes place in prokaryotes.
bacteria. So now,
Yes.
Lakshit, when you say binary fission takes place in prokaryotic cells, you will lose marks. You will get half out of 1. So binary fission, because amoeba is not prokaryote, it's a eukaryotic. So binary fission takes place in, so first difference is takes place in both prokaryotic and eukaryotic cells, eukaryotic organisms. Only in eukaryotic. Binary fission is basically it's a reproduction of new organism. This is related to growth or repair of an organism. This takes place only in unicellular. Only in multicellular. So mitosis, we had seen that the spindles were getting formed. Okay, so spindles are involved in cell division. No spindles. Example, bacteria, amoeba. Example, all multicellular eukaryote. So when the question comes, define binary fission. So the starting has to be, it's a type of asexual reproduction. When you write down asexual reproduction, now we differentiate between the mitosis and binary fission. In mitosis, it's a cellular division. That's what you start with. And in binary fission, it's a asexual reproduction, where a parent cell divides in two identical cells and thus produce offspring. बेटा ये जो दो सेल में डिवाइड हो रहा है, तो उन दोनों में यू कैन नॉट डिफरेंशिएट कि ये पेरेंटल सेल है और ये डॉटर सेल है. ठीक है. सो बेसिकली इट्स you can associate it with immortality also, right? So if something is dividing into two parts and then continuing dividing like that, so basically that particular cell is immortal in one way. Done.
Yes, sir.
Yes, sir.
Okay, here two questions can be formed. First question is, with the help of diagram, show binary fission of, number one is amoeba and number two is bacteria. How the binary fission will be different in both the cases, anyone? भाई अमीबा और बैक्टीरिया दोनों ही बाइनरी फिजन से मल्टीप्लाई कर रहे हैं, बट दोनों का डायग्राम अलग होगा। डायग्राम में क्या चीज, क्या पार्ट अलग होगा?
Pseudopodia, non-pseudopodia.
नहीं भाई, स्यूडोपोडिया नॉन स्यूडोपोडिया, स्यूडोपोडिया तो तब आएगा ना जब वो फूड के लिए है ना, न्यूट्रिशन इन एनिमल्स, उसमें डिफरेंसेस आएंगे। यहाँ पर स्यूडोपोडिया तो कोई काम ही नहीं है आपका डिवीजन में। रिप्रोडक्शन की बात करेंगे तो स्यूडोपोडिया हैज नो रोल टू प्ले। So what's the difference when you are drawing the diagram between bacteria and amoeba? No one, no one. I'm posting both the diagrams on the group. Now tell me, seeing the diagrams posted on the group, what is the difference?
So I think the cycle the उसके अंदर स्ट्रक्चर लगे अंदर का स्ट्रक्चर
Structure कौन सा structure भाई?
तो DNA
So what you will say, in amoeba there is elongation of nucleus, or basically nucleus is involved, while in bacteria there is nucleolus is there. So nucleolus is basically absence of nucleus is there. है ना, तो ये थोड़ा सा ध्यान रखेंगे when you are drawing the diagram for both. तो few students they tend to show bacteria में भी nucleus दिखा देते हैं and then they lose marks. Agreed on this?
Yes, sir.
So this is binary fission in amoeba is there. So you'll show it in 4 stage, and this diagram is there in NCERT and a popular question to come in exam. Very easy and simple question is there. Nothing to lose marks. You don't have to show here the organelles of amoeba or pseudopodia, nothing. You have to just show the nucleus and cytoplasm and then how the division is taking place. This binary division takes place separately in different organisms. So you can say in case of, let me first show you, then I'll write down. Here in case of amoeba, amoeba is spherical in shape, right? Amoeba is not spherical, but slightly spherical in shape. It has got certain volume also, and it divides into 2 parts spherically. When you talk about euglena, euglena divides, it also shows binary fission, but it divides longitudinally, it's dividing itself. When you talk about paramecium, paramecium divides itself transversely, right? This is transversely, paramecium is dividing itself. And then you have got, instead of binary fission, you have got multiple fission is there, where one parental cell divides into multiple cells, right? Before in binary, it's dividing into 2 cells. One cell divide into 2 cells. That's binary fission. If one cell is dividing into multiple cells, that is your multiple fission. Example is plasmodium. Anyone, which disease is linked with plasmodium?
Malaria.
Malaria, right? So it's a faster way of reproduction is there. So for spherical division, example is amoeba, longitudinal fission. What's the example for longitudinal fission?
Paramecium.
Longitudinal.
Euglena.
Euglena. Euglena is famous for what? Anyone? What's special about Euglena? No. नाइन्थ में अच्छा हमने मिस ही कर दिया ना, नाइन्थ का तो पूरा डाइवर्सिटी ही मिस कर दिया ना. यू हैव गॉट बेटा जो चेन ऑफ इवोल्यूशन है ना, चेन ऑफ इवोल्यूशन में यू हैव गॉट ब्लू ग्रीन एल्गीज आर देयर, देन यूग्लेना इज देयर. So Euglena is one of that very few organisms which shows both autotrophic and heterotrophic mode of nutrition. Euglena has got chlorophyll and Euglena is also heterotrophic. So Euglena is like ancestors in the chain of evolution. Euglena is ancestor for the plantae kingdom. So it shows both heterotrophic and autotrophic mode of nutrition. Transverse region is shown in
Paramecium.
paramecium. And multiple fission. Plasmodium. You can note it down. In this multiple.
Excuse me, sir.
Hanji beta.
Sir, yesterday I was going through NCRT and there is an in-text question which is asking why is DNA copying an essential part of the process of reproduction? Sir, इसका आंसर पता है लेकिन मुझे समझ नहीं आ रहा कैसे फॉर्म करूं.
Okay, when you say reproduction, it means that continuation of the species, and a species is being characterized by the number of chromosomes and the genes locked inside the chromosomes. So if number of chromosomes are changed or the genes or their sequence is changed, then the organism itself will change. You know, so thus first the DNA has to be copied and then the new cell will be produced. When you talk about an organism, then the characteristics of the organism related to characteristics and feature of organisms are guided by the genes which are there in the DNA. And for the continuation of the species, same DNA has to be copied first for same species to be continued.
Okay, thank you, sir.
ठीक है जी. So first one we have done is binary fission. Second type of asexual reproduction. Anyone?
Fragmentation.
Fragmentation. हमारा NCERT में सिर्फ दो ही mention है ना. One is your binary fission, second is budding. But we'll take some more because you never know, है ना. Some extra things always comes up. Basically, NCERT is a guide. You'll be surprised to know that CBSE and NCERT are separate departments. So CBSE recommend NCERT books as a guideline, but that's not 100% what is there. So something similar is there. You know, it has just given the syllabus. Syllabus mentions asexual reproduction, and for guideline you can use NCERT. That's what it says. Okay, first let's take the budding. What is budding? Anyone? One mark question. Define budding.
I think बिल्कुल एक new organism बनाना.
दोबारा बोलो. न्यू ऑर्गेनिजम्स बनाना. यार न्यू ऑर्गेनिजम्स तो सेक्सुअल में भी बनते हैं, तो बडिंग क्या हुआ? बडिंग तो आप यू आर जस्ट सेइंग इट्स अ टाइप ऑफ रिप्रोडक्शन. यू विल गेट 0 marks out of 1. Anyone else?
Sir, budding is the mode of reproduction in which there is an
ना गर्व देखो यहीं पे गलती कर दी ना. मोड ऑफ रिप्रोडक्शन फिर से बोलो. budding is a
Sir, budding is an asexual mode of reproduction in which there is
1 minute. 1 minute. Budding is asexual मोड ऑफ रिप्रोडक्शन। ठीक है, यहीं पर आपने अगर ये लिखा तो हाफ मार्क मिलेगा, और ये नहीं लिखा तो हाफ मार्क चला जाएगा। सिर्फ इतना लिखा तब भी हाफ मार्क्स मिल जाएगा, है ना। बडिंग इज अ मोड ऑफ रिप्रोडक्शन लिखा तो 0 मार्क्स। बडिंग इज एसेक्सुअल मोड ऑफ रिप्रोडक्शन, हाफ मार्क इज देयर। हाँ जी, आगे बोले। इन विच
Sir, there is an outgrowth in the parent's body which further matures and detaches from the parent body, becoming a new individual.
हाँ जी, वेरी गुड, इम्प्रेसिव। सो बडिंग इज अ asexual mode of reproduction in which, ठीक है, that's what difference is there between knowing and writing. तो 10th साइंस में है ना, till 9th साइंस I emphasize knowing is what is only thing required, है ना, you don't have to by heart it. But 10th साइंस you have to by heart. Same goes 11th, no need to by heart anything. ठीक है, थोड़ा लैंग्वेज में इधर उधर कर दिया कोई प्रॉब्लम नहीं, 1-2 मार्क्स कम आएंगे। 12th में again you have to by heart the things, you know, especially definitions. So budding is asexual mode of reproduction in which an offspring grows out of parent body and detaches itself to become independent. एग्जाम्पल क्या है भाई?
Hydra.
हाइड्रा,
Sir, yeast.
yeast. Okay, when I say give an example for asexual reproduction through budding in animals, ठीक है, then only answer will be hydra, है ना। yeast is not in animal kingdom. Yeast is not animalia, it is a fungi. So but simple अगर example पूछें, examples of budding, answer is hydra, yeast. Budding is shown in plantae also, so in that case it is cactus. How is, 1 mark question, how is budding different in hydra and yeast? How is budding different in hydra and yeast? I'm posting 2 pics on the group. Let me know the difference. One is for hydra, another is for yeast. Anyone? क्या हो गया भाई?
Sir, in budding in yeast, I think the nucleus of the parent is divided among the daughter cells.
No. In case of Hydra also, the first nucleus is being elongated and it's getting divided, and then the budding continues.
Sir,
Hanji.
sir, budding in yeast takes place very rapidly, so this forms kind of a chain.
Yeast, very good. Yeast shows multiple budding or chain budding. Anyone, how budding is different from binary fission? Major two differences I'm interested in. No one, no one. Chalo, I'll
Sir,
wait.
in binary fission, the parent cell divides into two daughter cells, and in budding, the splitting can vary from one cell to multiple cells.
Okay, so how fission is different from budding? So fission may you can have multiple fission is there, right? So budding may yeast can do multiple budding. Fission you can have binary fission. In Hydra you can have, you will have binary budding is there.
Sir, unicellular organisms reproduce through fission and multicellular through budding.
Very good, Pratna, impressive. But आपने थोड़ा सा गलत बोल दिया है. In case of fission, this is occurring only in unicellular, and in case of budding, yeast is unicellular, right? Hydra is multicellular. So both unicellular and multicellular. Okay, here the parental cell is not distinguished. पेरेंटल सेल आप बड़ी फिजन में बता ही नहीं सकते कि पेरेंटल सेल कौन सा है, ठीक है. और यहाँ पर जो पेरेंटल सेल है वो डिस्टिंग्विश्ड है ना, और यू कैन से डॉटर सेल is smaller than parental cell. So basically fission में you can say that concept of immortality is there, budding में that concept is not there. And then here you can say in example, fission example we know that's bacteria, amoeba. In case of budding, example is yeast, hydra. Right, let's take the third one. fragmentation. No, someone said fragmentation. Now please copy the differences, then we'll take fragmentation.
So copy.
Okay. One more question. Again, what is fragmentation? Hanjigar.
Yes, sir. So fragmentation is a mode of asexual reproduction in which a multicellular organism on maturation breaks up into many pieces, and each piece goes into a new individual.
You are getting half out of one. Anyone else?
So can I try?
Hanji bolo.
So a form of asexual reproduction where a parent organism breaks into many parts.
बेटा आपने तो सिर्फ ये बोल दिया पेरेंट ब्रेक्स इनटू मैनी पार्ट्स, दैट्स इट. मतलब आप कह रहे हैं कि एक जो है ना स्टार फिश जो है उसके अंदर मैं बबल मतलब हवा भर के उसको अगर मैं फोड़ दूं है ना तो बस हो गया, फ्रेगमेंटेशन हो गया. रिप्रोड्यूस पूरा डिफाइन नहीं हुआ ना, यू आर गेटिंग हाफ अ मार्क जस्ट बिकॉज यू सेड एसेक्सुअल रिप्रोडक्शन. Okay, let's take it. सबसे पहले तो mode of asexual reproduction in which the body of parent breaks into two or multiple pieces. ऐसा नहीं है कि हर एक में जो it has to be multiple pieces only. ठीक है, planaria में it breaks into two pieces and then each piece grows into a separate offspring, right? Into two or multiple pieces, and then you have to define that each of which grows and produce a new offspring. So it's something like if humans are showing fragmentation, then so once matured human is there, his arm will break off and that arm will slowly grow into a new human, right? Same thing is happening in case of a starfish. Starfish usually have 5 arms are there. So it sheds off 1, 2 or 3 arms and then its new arms will grow. So what happens is you have got a starfish is there. And let's say this starfish will shed off its arms. So this starfish has shed off 2 arms. And eventually this will start growing, this parental, it will start growing its new arms and this arms will start growing new bodies. And this will become your offspring. What are the examples for this? First I said was planaria. Second is
Spirogyra.
starfish. Spirogyra is for plantation, plantae kingdom. Na? and Hydra also shows this. In this, the splitting can be intentional or natural. Intentional or forced. So this organism planaria is there. This is what planaria looks like, right? This planaria, if you divide into two parts, each one will form one organism. So please show fragmentation. Let me know once copied.
Copy.
Okay, Pratna, let me know beta once you have copied.
So copy. Thank you.
Okay. Beta planaria has got a characteristics whereby using a knife, if you divide into two parts, you'll get two planaria. If you divide into 5 parts, you'll get 5 planaria. Each part will regenerate and will grow into a new planaria. You have understood this?
Excuse me, sir.
Hanji beta.
Sir, is there a possibility ki centuries after planaria, spirogyra, and all these things will turn into more complex animals?
Okay. When you talk about chain of evolution, okay, we all have undergone from evolution. Evolution in a sense कि अगर आप देखेंगे तो सबसे पहले एनिमेलिया में sponges were there, है ना? And these sponges evolved and they formed your jellyfish. Then jellyfish will evolve and they'll form something else. Now jellyfish does not have eyes, they don't have brain, they just have neurosystem, but they don't have any digestive system, nothing is there. Sponges का तो बिल्कुल ही कुछ नहीं है, न्यूरो सिस्टम भी नहीं है. Sponges को आप कट करेंगे, they will not feel any pain, है ना? Sponges are immotile. Thing is, during evolution, a part of it get evolved and remaining continued with the same species. ठीक है? Sponges जो है वो evolve होकर jellyfish बने, but ऐसा नहीं है कि sponges की existence बंद हो गई, नहीं. Some of the sponges evolved and they formed jellyfish, and then a new species of jellyfish is there and old sponges are there. Then again from jellyfish, part of it evolved and formed something else. You are getting my point?
Yes, sir.
So same way, starfish will evolve, you know, in centuries, you know, animalia, all these organisms keeps on evolving, but previously existing organisms remains there. So they say that the first organism to be to form on earth were arc bacteria, right? These arc bacteria, they can survive in acid, they can survive in volcano, you know, they can survive in extreme conditions, they can survive in space also. But they evolved, they formed bacteria which are prokaryote, they evolved to form eukaryote, but still the initial stages, they were still there. Okay. How is fragmentation different from regeneration? So when you talk about regeneration, regeneration is the regrowth is there. In case of fragmentation also there is a regrowth is there, but the part which has fragmented is also regrowing. खाली अगर आप रीजनरेशन की बात करेंगे तो जैसे सेलेमेंडर है, सेलेमेंडर की अगर बेटा इफ अ लेग इज कट, दैट लेग विल रीग्रो, रीजनरेट. बट ऐसा नहीं है कि जो लेग कट हुई है उसमें से नया सेलेमेंडर ग्रो करेगा. नो, अगर जो लेग कट हुई है वहां से नया सेलेमेंडर ग्रो कर रहा है, देन इट बिकम्स अ फ्रेगमेंटेशन. यू आर गेटिंग माय पॉइंट? स्टारफिश का इफ यू कट वन हैंड ऑफ द स्टारफिश, so from that hand also new starfish will grow, so then it becomes a fragmentation. और अगर सिर्फ कोई पार्ट रीग्रो करता है, then it's a regeneration, not fragmentation. Let me show you here the diagram. This is what I was talking about, planaria. You cut planaria into multiple parts. Each part will grow into new planaria. This is salamander is there. So this is what is regeneration is there. Right? It's not fragmentation, but it's only regeneration, because the leg which was cut, from this leg there is no new offspring is there. So we will not call it as fragmentation. Right? Agreed on this?
Yes, sir.
Okay. Fourth and the last one is parthenogenesis. Fourth one is parthenogenesis.
Regeneration कहाँ पर आता है फिर?
हाँ जी.
Regeneration.
Regeneration, that's what I was saying. Regeneration is not mode of reproduction.
Oh.
Regeneration is when a part of the body is growing back, it's regeneration.
Okay.
So parthenogenesis, it is form of asexual reproduction. Hanji, anyone? What happens in this parthenogenesis? Okay, write down where female egg can reproduce offspring even in absence of male gametes.
सर एनसीईआरटी में रीजेनरेशन को एक अलग ही रिप्रोडक्शन का मोड देके समझाया है.
दोबारा बोलो.
सो एनसीईआरटी में फ्रेगमेंटेशन के बाद रीजेनरेशन है और वो अलग से एक एसेक्सुअल मोड का रिप्रोडक्शन का मोड बताया है.
अच्छा जी, लेट मी चेक आउट ऑन दिस. ठीक है, रीजनरेशन देखो, व्हेन देयर इज अ रीजनरेशन ऑफ बोथ द बॉडी पार्ट्स, राइट, देन वी कॉल इट एज फ्रेगमेंटेशन. When but only regeneration is not reproduction, you know. Let me just check out and then I'll confirm on that what NCERT is saying.
Okay, sir.
But I hope you are getting my point, what I'm trying to convey here.
Yes, sir.
Just a second. ओके बेटा, उसमें से आपने लास्ट लाइन पढ़ी आपने. इट सेज however regeneration is not same as reproduction since most organisms would not normally depend on being cut up able to reproduce. Okay, but I think it's saying planaria is reproducing by regeneration, it's saying. ठीक है जी. We'll take it what NCERT is saying, you know. So NCERT is not naming this as fragmentation, it is naming this as regeneration. Okay, where each of the body part is getting regenerated into a new offspring. ठीक है, तो इसको थोड़ा सा आप चेंज कर लें. अच्छा, बट फ्रेगमेंटेशन तो सेपरेट लिया ही हुआ है उसने. Fragmentation it is taking only for Spirogera, and regeneration it is taking for planaria and this thing. So okay, this is Spirogera को हम अलग से कर लेंगे, है ना. इसको regeneration कर लेते हैं. And fourth part we will take it is fragmentation. Fragmentation we will take Spirogera. And this will take it as fifth part. ठीक है, but planaria can split itself intentionally also, है ना. ऐसा नहीं है कि it needs someone to cut it into two parts. It can divide itself intentionally also in two parts. Okay, parthenogenesis. So in this, what's happening is, let's say in case of a bee, you have got a queen bee is there. Now only the queen bee is laying the eggs, है ना. No other bee in the hive is laying the eggs. So queen bee, when it's laying an egg and it's an unfertilized egg is there, unfertilized eggs. Okay, there is no male gametes are involved. These unfertilized eggs will develop and they will form male offspring. This is haploid. And if the queen bee has mated with the male bee, then the egg will be your fertilized egg. This will be diploid. And here female will be produced. This female will be, most of them are workers. These are your drones. And one of them will again become a queen. So this is B is showing asexual. This is B is showing sexual reproduction. So it's showing both sexual and asexual. This asexual is what we'll call it as parthenogenesis. Parthenogenesis.
सर एक ही सेक्स क्रोमोसोम की हेल्प से हम ये कैसे डिसाइड कर सकते हैं कि जो ऑफस्प्रिंग है वो मेल होगा एसेक्सुअल रिप्रोडक्शन में?
तो बेटा बी का जो स्ट्रक्चर है दैट इज like that only, that if the same set of cells are there, I mean, basically what happens is जो बी में जब जाइगोट डिवाइड होता है तो डिवाइड होने के बाद जो 2 सेल्स बनते हैं वो दोज 2 सेल्स फ्यूज टुगेदर and then they form the pair of chromosomes. But in that the pair of chromosomes are exactly same. So agar same set of pair of chromosomes rehte hain, then a male offspring will be produced. And if the egg is fertilized, then basically one set of chromosome are from male, one is from female, and the diploid cells are produced and it forms a female is produced.
Okay, sir.
ठीक है. So for example, let's say the hive is there. Usually hive कैसे produce होती है बेटा? So in a beehive there is a queen and lots of workers are there. Lots of female workers are there. Now, if it's already the colony is already getting overloaded with number of workers, then from that colony one queen will leave with half of the workers. तो जो queen जो leave कर रही है वो half of the workers means सब females के साथ ही leave करेगी. फिर then that queen when it's laying egg, तो starting के जो egg हैं those will be all unfertilized eggs. From that male offspring will be produced, right? This male offspring will fertilize the queen egg now, and they will form fertilized eggs, and then female bees will be produced. ठीक है. जो हाइव होता है उसमें जो ड्रोन्स हैं जो मेल बीज हैं, देयर जॉब इज ओनली टू फर्टिलाइज फीमेल एग, दैट्स ऑल, नथिंग एल्स.
Okay, sir.
ठीक है जी. आई होप यहाँ पे ये क्लियर है. ठीक है. रीजनरेशन इन जनरल द वर्ड इज वेयर द बॉडी पार्ट इज गेटिंग रीजनरेटेड, but NCERT has taken it that regeneration is mode of reproduction whereby the parts which is cut are regenerating into new offsprings. ठीक है. Fragmentation is where the body part is dividing into multiple parts by its own. An example is pyrogera. And the last one is parthenogenesis. Then in this last is what we'll take it as cloning. Okay, sixth one you can take is cloning. Cloning is also asexual, but it is artificial asexual reproduction. Okay. So in cloning, what's done is the egg cell is taken from a mother. Let's say from A, the egg cell is being taken. From that egg cell, the nucleus is removed, right? So the nucleus is removed. Now this egg remaining part is being left. This part is basically is high in nutrition and with characteristics of dividing itself. That characteristics is there, but there is no nucleus in it. So what they do is from another organism, from the donor, they take a healthy cell. From that healthy cell, they take a nucleus. With the help of, now this nucleus is being implanted inside the egg cell. ठीक है. तो आपका this egg cell is there. Initially it was having a nucleus with haploid. That means one set of chromosome was there. So that nucleus is removed and the healthy nucleus is being put. This nucleus is having two sets of chromosomes are there, right? Pair of chromosome is there. Now with the help of electricity, this cell, egg cell is stimulated to start dividing. So when this egg cell starts dividing, then it will keep on producing new cells, and then it will form a new organism in which this nucleus is getting duplicated every time. So the clone of B is getting produced here, right? Once it forms a blastocyst, this blastocyst is being put back again in organism A. You're following this? So beta cloning mein what they are doing is, let's say they will take a, this is a black sheep, this is a white sheep, right? So black sheep ka jo hai, they are taking the egg cell. उस egg cell में now they will remove the nucleus, है ना? इस egg cell का जो है nucleus जो है they will remove. The nucleus of egg cell is removed. They will take a healthy donor cell from a white sheep. White sheep से एक donor cell लिया, ठीक है? उस donor cell का जो nucleus है, that nucleus is implanted inside the egg cell. This egg cell is being stimulated. ये stimulate होता है तो then it starts dividing itself and it behaves as an embryo. This blastocyst which is formed, this is implanted again in the mother, जिससे egg cell लिया था, ठीक है? And then the baby starts growing in the mother, and the child which is produced is having exactly same set of DNA, you know, same set of DNA without any change. And the sheep which is produced here is clone of that white sheep. The black sheep is giving birth to a white sheep. Understood this?
Yes.
That's what cloning is. So cloning of animals is banned in half of the countries. Cloning of humans is theoretically banned throughout the world, but few countries are working on it unlawfully. So that's the end of chapter. Any doubt you have, or we'll start with construction? Construction start करें बेटा?
Yes, sir.
कंस्ट्रक्शन में हमारा टेंजेंट्स ड्रॉ करने का रह गया है ना, तो टेंजेंट्स में आपके पास जो क्वेश्चन आते हैं, हाँ जी,
No, sorry, sir, nothing.
नथिंग, ओके, सो ड्रॉ टेंजेंट है ना, ड्रॉ टेंजेंट में जो क्वेश्चन आएंगे यहाँ पर आपको इट इज सेंटर is given, center not to be used, ठीक है, and then point is on circle, point is outside circle. तो बेटा ये 4 टाइप के क्वेश्चंस आपके आएंगे, प्लस इसमें एक दो वेरिएशन है जहां पर एंगल बिटवीन द टेंजेंट्स इज गिवन, ठीक है. सो दिस इज क्वेश्चन नंबर 1, क्वेश्चन नंबर 2, क्वेश्चन नंबर 3, क्वेश्चन नंबर 4. So first question is point on circle and center given. So this is the center of the circle is given and this is the point on the circle. You have to draw the tangent from the point P on the circle. कैसे draw करेंगे बेटा? हाँ जी, number 1, draw circle. Number 2, join OP. ठीक है, आपको पॉइंट P पर टेंजेंट ड्रॉ करनी है, so you will join OP. Number 3, draw 90-degree angle at P. Number 4, draw tangent. ठीक है, प्रैक्टिकली आप क्या कैसे करेंगे, आप पहले यहाँ पर विद द हेल्प ऑफ प्रोटेक्टर यू विल ड्रॉ 90-डिग्री एंगल, है ना, आप प्रोटेक्टर रख के 90-डिग्री एंगल यहाँ पर ड्रॉ करेंगे, बट देन यू विल शो इट एज इफ यू हैव ड्रॉन विद द हेल्प ऑफ कंपास, आपने कंपास की हेल्प से ड्रॉ किया है। ठीक है, शो इट दिस एंगल इज 90 डिग्री। इजी था।
Yes, sir.
एक बार क्या किया आपने?
बेटा हमें एक सर्कल गिवन है, सर्कल पे पॉइंट गिवन है, वहां से हमें टेंजेंट ड्रॉ करनी है, हमें साथ में जो है सेंटर ऑफ द सर्कल गिवन है। तो अगर सेंटर ऑफ सर्कल गिवन है तो आप सेंटर और उस पॉइंट को ज्वाइन करेंगे, है ना, दैट इज O एंड द पॉइंट P इज गिवन टू यू। So you will say given is given point O and point P, ये given है आपको। अभी क्लियर है बेटा आपको?
यस सर।
हाँ जी, just a minute ना, let me post it on the group also. ओके, नेक्स्ट वन। यहाँ पे क्या था, हमारे पास सेंटर गिवन था, ठीक है। जो नेक्स्ट क्वेश्चन आता है, या तो आप सेंटर जो है फाइंड करेंगे, लेट मी टेक द नेक्स्ट क्वेश्चन फर्स्ट।
तो वो वाले क्वेश्चन कैसे करना है जिसके अंदर बोलते कि ड्रॉ 5 सेंटीमीटर लाइन पे?
हाँ, डोंट वरी, आई विल टेक ऑल ऑफ देम, है ना। So next question is find center of circle. Find center of circle and draw tangent from point P. भैया एक सर्कल गिवन है, इसमें ये पॉइंट P गिवन है, वो कह रहे हैं आप पहले सेंटर ऑफ सर्कल फाइंड करें और उसके बाद पॉइंट P से टेंजेंट ड्रॉ करें। सेंटर ऑफ सर्कल कैसे फाइंड करेंगे बताएं फटाफट।
सर दो कॉर्ड्स बना के उनके परपेंडिकुलर बाइसेक्टर्स।
दो कॉर्ड, ठीक है, उन दो कॉर्ड्स के परपेंडिकुलर बाइसेक्टर्स, एंड उसके बाद सेम एज क्वेश्चन 1। एग्रीड ऑन दिस?
Yes, sir.
Number 1, draw two chords AB and BC. Draw perpendicular bisector of AB and BC. Intersecting point is center of circle, and then follow steps of question 1. ठीक है, मुश्किल से देयर 7-8 क्वेश्चंस आर देयर, है ना, उसी में सभी कवर्ड हो जाएंगे, ईच एंड एवरी वन विल बी कवर्ड। Question number 3. draw tangent on a circle with point P on it without using center of circle. आप सेंटर ऑफ सर्कल यूज ही नहीं कर सकते बेटा, उसको यूज किए बिना आपको जो है टेंजेंट ड्रॉ करनी है। हाँ जी, बताइए कैसे करेंगे। टिंग 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 टिंग, आई एम वेटिंग। हेलो जी, क्या हो गया?
सर एक एक्सटर्नल पॉइंट लेंगे और फिर उससे ज्वाइन कर देंगे।
ना
तो
ना, कोई एक्सटर्नल पॉइंट नहीं लेंगे, ये तो कॉमन क्वेश्चन है यार, कराया नहीं है स्कूल में।
No, sir.
ओके, सो फ्रॉम पॉइंट P ड्रॉ अ कॉर्ड, है ना। यहाँ पर मैं बेटा आई एम यूजिंग योर कॉर्ड टेंजेंट थ्योरम, है ना, कॉर्ड टेंजेंट थ्योरम हम यूज करेंगे। सो मैंने पॉइंट P से कोई एक रैंडम कॉर्ड ड्रॉ की है PA। फ्रॉम PA आई एम ड्रॉइंग अ रैंडम एंगल, कोई भी रैंडमली एक एंगल ड्रॉ कर लें आप, PB, ठीक है। इस एंगल को आपको कॉपी करना है। कहाँ पे कॉपी करना है? यहाँ पे कॉपी करना है। एंगल कॉपी करना आपको आता है। बी से जो है आप एक आर्क, कंपास ओपन करें, बी पे नीडल रखेंगे और ये आपने आर्क ड्रॉ किया। सेम वे पी पर आप नीडल रखें एंड ड्रॉ दिस आर्क लाइक दिस, है ना, ये आर्क आपने ड्रॉ की। उसके बाद यू कीप योर नीडल एट दिस पॉइंट एंड यू मेजर हाउ मच इज दिस। कॉपिंग द एंगल कर रहा हूं मैं, है ना। कंपास की नीडल आप यहाँ पर रखें, सेम कंपास ओपन रखें एंड यू कट दिस आर्क हियर। तो मैंने बेसिकली एंगल कॉपी किया है एंड यू जॉइन दिस PM। PM is what is the tangent. So first step will be from point P, randomly draw chord PA. From PA, draw triangle PAB with point B on circumference. Copy angle B at point P. Draw tangent. ये आपको समझ में आया? टेंजेंट कॉर्ड थ्योरम वी हैव यूज्ड फॉर दिस। बेटा अगर
Yes,
डाउट
yes.
है तो आई विल टेक वन मोर क्वेश्चन ऑन दिस।
सर हम रैंडमली कोई भी ट्रायंगल ले सकते हैं P को एक पॉइंट लेकर।
हाँ जी, रैंडमली आप P से, पॉइंट P से कोई सा भी ट्रायंगल ड्रॉ कर लें, जैसा मर्जी ट्रायंगल ड्रॉ कर लें। तो बेसिकली P से आप पहले एक कॉर्ड ड्रॉ कर लें PA, फिर उसके बाद पॉइंट B ले लें। आप इस चीज को समझें कि अगर मैंने ये बी यहाँ लिया होता तो ये एंगल जो है, ये एंगल तो सेम होते ना। नाइन्थ की थ्योरम में कॉट सबटेंड द इक्वल एंगल ऑन द सेम साइड ऑफ द सेगमेंट,
Yes, sir.
है ना। तो सेम एंगल यहाँ कॉपी हो रहा था। और यहाँ हमने कौन सी थ्योरम यूज किया है कि एक सर्कल में अगर हम ये टेंजेंट ले लें और ये कॉर्ड ले लें, तो दिस एंगल बिटवीन द टेंजेंट एंड द कॉर्ड इज सेम एज द एंगल सबटेंडेड बाय द कॉर्ड। ये दोनों एंगल कैसे होंगे? इक्वल होंगे।
Okay, sir.
चेक करें, स्टिल एनी डाउट? डन। Okay, next one. From a point P outside the circle with center given, draw the tangents. ठीक है ना, यहाँ से दो टेंजेंट्स ड्रॉ करनी है आपको सर्कल पे। ठीक है, वन थिंग इज देयर कि जो टेंजेंट है और जो रेडियस है, दे आर परपेंडिकुलर टू ईच अदर। ठीक है, इस फैक्ट को हम यूज करेंगे। विल सी डिफरेंट रेडियस पे आपको अगर परपेंडिकुलर ड्रॉ हो जाती है लाइन, देन इट बिकम्स अ टेंजेंट। एनीवन हाउ टू डू इट? भाई सेंटर ऑफ सर्कल गिवन है आपको, ठीक है। सो आई विल जॉइन P एंड O। ठीक है, अब यूजिंग PO एज अ डायामीटर, अगर मैं यहाँ पर सर्कल ड्रॉ करूंगा ना, मैं पहले सिर्फ आपको समझा रहा हूँ ना, रीजनिंग आई एम टेकिंग। अगर मैं यहाँ पर डायामीटर यहाँ पर ड्रॉ कर रहा हूँ, ठीक है, नाउ थिंक ऑन इट। सिर्फ अगर मैं इस सेमी सर्कल को देखूं बेटा, तो सेमी सर्कल में डायामीटर ऑलवेज सबटेंड्स एन एंगल ऑफ
90 degree.
90 डिग्री। कहीं पर भी मैं बनाऊं तो 90 डिग्री का एंगल बनेगा। ठीक है,
Yes, sir.
तो अगर मैं यहां पर बनाया ये एंगल और ये एंगल कितना डिग्री का होगा?
90.
90 डिग्री। ठीक है, ओ ए जो है वो आपका रेडियस है, देन पी ए हैज टू बी द टेंजेंट, इजंट इट?
Yes, sir.
So what steps will be there is, so point P was given to me outside the circle, center of the circle O is also given to me. Agreed? So I have to draw a circle with PO as a diameter. PO as a diameter, I have to draw a circle on it. तो बेटा सर्कल ड्रॉ करने के लिए सबसे पहले सेंटर फाइंड करना होगा। PO का सेंटर कैसे फाइंड करेंगे? Anyone?
Perpendicular bisector.
परपेंडिकुलर बाइसेक्टर ऑफ PO। परपेंडिकुलर बाइसेक्टर के लिए आप पहले P पे नीडल रखें, ओपन द कंपास मोर देन हाफ, ओपन नीडल रखें, ओपन द कंपास मोर देन हाफ एंड join these two. What will be this point? This point is O dash, which is center of PO. You keep the needle at O dash and draw a circle. Name the intersecting point as A and B. Join PA. ठीक है, PA and PB are the tangents. अब यहां पर प्रॉब्लम कहां पर है? आपके नंबर क्यों कट गए?
सर 90 डिग्री मार्क नहीं करा।
कहाँ पर मार्क नहीं किया? क्यों मार्क करने की जरूरत है? जब जरूरत यहाँ पर जहां जरूरत थी वहां पे मैंने मार्क कर दिया। एक्सप्लेनेशन तो उसने पूछी नहीं है। नंबर इसलिए कट गए क्योंकि जो टेंजेंट है वो लाइन सेगमेंट नहीं होती, टेंजेंट लाइन होती है आपकी, है ना? लाइक अ सीकेंट, टेंजेंट इज अ लाइन, नॉट लाइन सेगमेंट, सो इट हैज टू बी लाइक दिस। समझ में आ रहा है?
यस।
हाँ जी तमनजोत, क्लियर है बेटा?
Yes, sir. Clear है.
So here the first step will be join PO, draw perpendicular bisector of PO and find its center O dash with PO dash as radius and O dash as center. Draw a circle. Name two intersecting points of two circles as A and B. Join PA and PB. PA and PB are required tangents. हाँ जी डन।
यस सर।
ठीक है, अब इसमें वेरिएशन कैसे आ जाता है? क्वेश्चन इज circle of radius 5 centimeter is given with center O. Draw tangents on circle making an angle of 60 degrees. ठीक है, अगर आप यहाँ पे पहले एक रफ फिगर ड्रॉ कर लें इट विल हेल्प यू आउट। तो बेटा ये एक सर्कल गिवन है हमें, सर्कल का जो रेडियस है वो 5 सेंटीमीटर है, सेंटर ओ है। अब इस पे दो टेंजेंट्स आपको ड्रॉ करनी है, उनके बीच में एंगल जो है मेकिंग एन एंगल ऑफ 60 डिग्री विद ईच अदर, उनके बीच में एंगल 60 डिग्री बनना चाहिए। तो क्या करेंगे? एनीवन? मैं आपको रफली वैसे बता देता हूँ, है ना भाई। यहाँ पर जो हमारे पास ये जो बेटा टेंजेंट्स बनेंगी, है ना, ये ऐसे टेंजेंट्स बनेंगी। अब इनके बीच में ये एंगल बोल रहा है वो 60 डिग्री होना चाहिए। तो यहाँ से अगर मैं रेडियस ड्रॉ कर देता हूँ तो रेडियस एंड टेंजेंट्स आर परपेंडिकुलर, तो ये तो 90 है, ये भी 90 है, तो ये एंगल कितना डिग्री हुआ हमारा?
120.
120। तो बस उसी को लेकर हम प्रोसीड करेंगे। ठीक है, तो हम उल्टा चल रहे हैं यहाँ पर। उल्टा कैसे चल रहे हैं? सबसे पहले आप यहाँ पर कोई भी एक रैंडमली रेडियस बना लें। ठीक है, इसको नाम दे दें ए। यहाँ से हमारी एक टेंजेंट जाएगी। अब यहाँ पर ये जो एंगल है ये कितना डिग्री होना चाहिए था? 120। तो आप कंपास यूज करें और ये आपने एक आर्क, ये 60, ठीक है, ये 60 डिग्री, ये 60 डिग्री, एंड दिस इज व्हाट यू विल ड्रॉ। ये समझ में आ रहा है? आपने 120 डिग्री ड्रॉ किया है यहाँ पर। दिस
Yes,
इज बी, दैट्स इट। अब यहाँ पर आप 90 डिग्री का एंगल ड्रॉ करें। ठीक है, आप चाहें तो प्रोटेक्टर यूज करें, बट यू शो इट एज इफ यू हैव यूज्ड कंपास टू ड्रॉ दिस। ठीक है, इसको आप ज्वाइन कर दें और दोनों साइड एक्सटेंड करें ना, टेंजेंट है ये। सेम वे पॉइंट बी है ना, ये आप एंगल बनाएं यहाँ पर। सो 60 60 एंड देन बाइसेक्टर ऑफ दिस इज 90 एंड दिस इज द टेंजेंट यू विल ड्रॉ। दिस एंगल इज 90, ये 120 है, तो जो हमारा ये एंगल बनेगा ये कितना डिग्री होगा बेटा?
60.
60 डिग्रीज। Right, step 1 will be draw a circle with radius 5 centimeter. Randomly draw radius OA. With OA as base, draw angle 120 degree at point O. Okay. With second arm OB, where OB is also radius. From point A and B, draw 90 degree angle and thus tangents. These tangent will make an angle of 60 degree with each other. ठीक है जी। आपको अगर कोई कंस्ट्रक्शन का क्वेश्चन है जिसमें आपको डाउट है तो आप प्लीज उसको मुझे फॉरवर्ड करें। आई टेक दैट क्वेश्चन ऑन मंडे बिफोर साइंस क्लास।
Okay, sir.
Okay, sir.
ओके, चलो जी, देन लेट्स एंड द सेशन।
तो बस इतना ही था।
बस इतना ही है।
ओके सर।
Thank you, sir.
ओके, वेलकम।
थैंक यू सर, बाय सर।
वेलकम बेटा, बाय बाय।